Prophase. It's the first and longest phase of mitosis, in which the genetic material, the chromatin inside the nucleus, condenses and the chromosomes become visible. You can see an example of prophase in a plant cell.
Isn't there anyone who knows what it's all about? Sure, Charlie Brown. I can tell you what it's all about.
The cell cycle divides the cell life into two parts, interphase and division. Interphase makes up 90% of the cell's life. It is divided into three parts. The first part, gap 1, involves protein synthesis. It keeps the cell growing. Once the cell reaches the G1 checkpoint, the decision to divide is made. During the S, or synthesis phase, DNA replication occurs. Then the last phase, gap 2, prep for division occurs. By then, all the organelles have replicated and the centriole pairs are present. The nucleus and the nucleolus are also present. Mitosis is somatic nuclear replication, otherwise known as the dividing of the nucleus. When coupled with cytokinesis, it produces two cells that have the same ploidy as the parent. These cells are known as diploid cells. The DNA is identical in the daughter cell as the parent cell. Mitosis is used for the growth and maintenance of the organism. During prophase, the chromatin condenses into visible chromosomes containing two sister chromatids held together by cohesin proteins. The asters begin to migrate towards opposite poles of the cell as the mitotic spindles form. Prometaphase is the second stage of mitosis. The nuclear envelope degrades and the chromosomes are more condensed. The asters are now at two opposite poles of the cell. The mitotic spindles stretch across the cell. Two types of microtubules make up the spindle: kinetochore microtubules, which attach at the kinetochores of the chromosomes, and non-kinetochore microtubules that interact with those from the opposite pole. Metaphase is the third step in mitosis. The chromosomes have now aligned along the metaphase plate, an equatorial plane dividing the cell in half. Anaphase happens next. It is the shortest stage of mitosis. The cell begins to elongate as the non-kinetochore microtubules push against each other. The kinetochore microtubules pull on the cohesins and separates the sister chromatids, ending with equivalent and complete sets of chromosomes. The last phase of mitosis is telophase. Two daughter nuclei form, and the nuclear envelope and nucleolus reappear. The microtubules break down, and finally we end with two diploid cells. Most times, telophase and cytokinesis, which is the division of the cytoplasm and separating of the two cells, occur simultaneously. In animal cells, a cleavage furrow, which is a contractile ring of microfilaments, splits the cells in two. In plants, vesicles form a cell plate which becomes the new cell wall.
Meiosis immediately follows G2 of interphase. Chromosomes have been duplicated before the cell even begins to divide. Meiosis has two parts, each with 4 stages: meiosis 1 and meiosis 2. Cytokinesis occurs twice, once for each part. The starting cell is diploid. In prophase 1, duplicated chromosomes condense. The nuclear envelope breaks down, and as a result, the nucleolus disperses. The microtubule organizing centers migrate to opposite poles of the cell as the mitotic spindle forms. Kinetochores form at the centromere of each condensed duplicated chromosome. They attach to the kinetochore microtubules. Synapsis occurs during prophase 1. Homologous chromosomes pair over their entire lengths except the centromere, forming tetrads. Crossing over may occur. Locations of crossing over are called chiasmata. In metaphase 1, kinetochore microtubules grow and shrink to line up duplicated condensed chromosomes, still paired as tetrads, at the metaphase plate. Tetrads are arranged with each chromosome in a pair on either side of the metaphase plate. This independent assortment occurs randomly, based on the location of the chromosomes as they condense and pair. In anaphase 1, tetrads are pulled apart as each duplicated condensed chromosome in the homologous pair moves to opposite poles of the cell. The same process that occurs in telophase and cytokinesis of mitosis also happens in telophase and cytokinesis 1 of meiosis 1. However, it produces two new haploid cells with different DNA from the parent cells. Meiosis 2 is similar to mitosis. However, in meiosis 2, two haploid cells divide to create 4 haploid cells. The 4 haploid cells are generally not identical to one another. It results in the creation of new genes and a combination of genes.
Independent assortment and crossing over cause genetic diversity and allow for the continuation and evolution of a species.